is genetics the ultimate ruler for us, or the decider for us? Is the genetics the big boss, or do we get a say in it? Ladies and gentlemen, good afternoon, my dear teachers, friends and colleagues. I'm Dulang Ratnayaka, Senior Registrar in Cardiology in Kandy. So before I start off, let's just recap a few things. So we know from faculty times that DNA is made of a sugar phosphate backbone, attached to which is a nitrogenous base, namely adenine, thymine, guanine, and cytosine. And these DNA strands are coupled with each other and they're twisted into a helix. And we know that these DNA just don't lie around. This DNA is wound around a protein called histone, tightly wound around a protein called histone. This DNA histone complex is called the nucleosome, and these nucleosomes are compact into chromosomes. Right. So we know that early fetal life is, it starts with one cell, divides into 2, 4, so on and so forth. And at a certain point, it goes into what we call a blastocyst. Now, this blastocyst has absolutely identical, phenotypically and genotypically identical cells. However, now these cells go into different types of cells, like the heart, the liver, and the brain, which has completely different phenotype and function, maintaining absolutely identical genetic material. How is this possible? Now, who does this? This actually happens because certain parts of the DNA are switched off or switched on when it goes into different organs. So who switches this on and switches this off? This is where epigenetic comes to play. Epi in Greek meaning above. So what we are going to talk about is something above genetics. So what exactly is epigenetics? It's a molecular bridge between genes and the environment. There are multiple definitions, but there are 3 common definitions that have been used in the scientific world. The one definition is the transmission and perpetuation of information. All of these are cells have certain information. This information is transmitted and kept on multiplying, but this information is not actually in the DNA. And this information is multiplied through meiosis and mitosis. Another definition is that it's mitotically and meiotically heritable changes in gene expression that are not coded in the DNA sequence itself. Kind of similar, but this is the definition that has been used commonly in cancer research. The third definition that I would like to point out is the mechanism for stable maintenance of gene expression changes that involves physically marking DNA or its associated proteins, which allow genotypically identical cells to be phenotypically distinct. Now, this is what happens when we talk about the blastocyst multiplying into different organs. So simply saying, epigenetic pathways regulate gene activity in an inheritable manner without changing the nucleotide sequence of the DNA. So through this process is what allows us right. So our genetic material, these chromosomes have genes, and this genetic material have the ability to turn on and off genes from time to time. This turning on and off is what really decides how the blastocyst goes into different types of organs. Our genetic material are in active form or repressed form, what we call active chromatin or repressed chromatin. So when the genetic material is active, a part of the gene is active, the DNA is open and it allows the RNA polymerase to act on the DNA, producing RNA and thereby producing different types of proteins. But if the DNA is repressed or the DNA is closed, then this RNA polymerase cannot act on it and mRNA is not produced. So how does this genetic material open and close, or they become active or repressed? This is through several mechanisms, and these mechanisms can be due to DNA modification or the histone modification. DNA modification is actually mainly due to DNA methylation. Histone modification, the two most common modifications are acetylation and the methylation. So let's just get into a bit of technicalities with this. So when we talk about DNA methylation, what actually happens is a methyl component comes and binds the cytosine ring of cytosine base of the DNA, and it doesn't happen anywhere else. It just doesn't happen all over the place. It just happens in the 5' location of the cytosine ring, and depending on where this methylation occurs, this attaching of the methyl part occurs, the activation or the deactivation of the DNA is decided. Like for example, when the methylated sites are located in the promoter genes, it suppresses the gene, whereas this methylation occurs within the gene body, it increases or it activates the gene. Likewise, as I mentioned before, there are changes that occurs into the histones as well. Now, histones have this chemical projections, what we call histone tails. Now, these histone tails can be attached by different types of chemical compounds with use of enzymes, and thereby activating the gene or deactivating the gene. Now, this histone tail is actually positively charged, while the DNA is actually negatively charged. As we know, as the opposites attract, the histone, because of these positive and negative charges, the DNA is tightly bound to the histone. What if we can change the charge of the histone protein? What if we can make the positive charge less positive or make it negative? Then as negative repulses, the tight bond between the DNA and the histone will be loosened up, opening up the DNA. So that's what exactly happens when it comes to acetylation. When an acetyl component comes and binds to the histone tail, the positive charge reduces and the bond between the DNA and the histone loosens up, opening up the DNA. So simply saying, acetylation opens up the DNA, and the deacetylation closes up the DNA, causing the gene to be repressed. Like acetylation, methylation also can occur in the histone tail, but depending on where the methyl component comes and binds, whether the gene is active or repressed depends. If it's attached to this lysine 4 component, it activates the gene, while when it attaches to lysine 9 and 27, it represses the gene. So methylation, and depending on where it's methylated, acetylation and even phosphorylation activates or deactivates our chromatin. Right. So now we know the mechanisms, but what is in it for us? Why is a cardiologist talking about epigenetics? Now, I'm sure as clinicians, all of you have come across being ischemia, second being diabetes mellitus. And we obviously have come across patients where we were not able to identify a cause, where we label them as idiopathic. Or is it actually idiopathic? Multiple epigenetic modifications have been identified to be associated with dilated cardiomyopathy. So this could be the missing link that we are that's causing this idiopathic dilated cardiomyopathy. And a team in 2008 have knocked off a particular changed the epigenetics of a particular genome in rats and found out that it rapidly progresses the dilated cardiomyopathy. What about hypertension? Now, as clinicians, we manage hypertension day in, day out. But what we actually do is just control the condition. Are we actually addressing the problem? Do we know what actually causes this essential hypertension? What causes the vessels to be this stiff, which results in essential hypertension? Studies have identified the increased expression of alpha-1 receptor because of certain epigenetic change in rats. And also, this second point, it's very important, where maternal low-protein diet during pregnancy has led to reduced methylation of a promoter gene of a particular gene, which eventually has caused hypertension in the offspring in adult life. So this epigenetics could be actually the missing link. Then talking about atherosclerosis, as clinicians, I'm sure you have seen patients who have modest amounts of or moderate levels of LDL, moderate levels of cholesterol, but they have serious atherosclerotic plaques, or they end up getting ST-elevated MI or non-ST-elevated MI. How is this possible? So, and also, as cardiologists, what we have noticed is we cannot really identify why a certain atherosclerotic plaque, what makes the atherosclerotic plaque unstable. We don't know exactly what makes the atherosclerotic plaque unstable. Sometimes we have seen patients with moderate-sized atherosclerotic plaque causing ST-elevated MI or non-STEM. The link, what they have found that there is a connection between this dysfunctional epithelium or the unstable plaque, and there's a connection between epigenetics and this dysfunctional epithelium. Same goes to diabetes mellitus. In patients with diabetes mellitus, obesity, and related metabolic conditions, there have been serious epigenetic changes involving in multiple, in different ethnicities. So epigenetics is involved in many non-communicable diseases, way deeper than we know, or probably even a lot more than we expect. And I believe that we don't give epigenetics enough prominence as clinicians. So let me ask these questions again. Can we alter our own epigenetics? Or what alters the epigenetics? Are epigenetics inherited? To answer these questions, let me give you a small history lesson. In 1944 to 1945, there has been a serious famine in Netherlands, where Germans have cut off routes of supply routes to Netherlands, and Netherlands has gone into a serious famine, and the authorities had to ration out the food supplies to the Dutch nationals, and at a certain point, these people were living on just potato and bread. And this has led, this event has permitted researchers to examine long-term consequences of famine. And what they have found out was that the studies have strongly supported in utero exposure to famine may have resulted in adverse metabolic and mental phenotypes, which depended on sex and the timing of the exposure during the gestation. And this study has shown that a sex-specific increased risk of obesity, dyslipidemia, and cardiovascular-related deaths has been observed 6 decades later among those whose mothers were exposed to the famine during the early gestation. Now, this shows that environmental change have altered the epigenetics of the fetus. And this study has been taken further by Heijman and his colleagues, to where they have identified serious methylation, different methylation patterns in this in the population which has been exposed to this famine than as opposed to the controls. So what's the lesson here? What we can get from this is that our environmental changes, our behaviors may alter our epigenetics, and they might cause adverse results in the future. This leads to a very interesting hypothesis, what we call the Thrift gene hypothesis, where an exposure to an hardship leads to epigenetic change in certain alleles, which will allow them to survive during these hardships. Generations down the line, when the conditions are favorable, this may result in adverse outcomes. Like, for example, short-term epigenetic adaptations that take place shortly after conception, like getting exposed to a famine shortly after during the early fetal life, causes epigenetic changes in the fetus, which allows them to survive during these hardships. However, when these children are born and are exposed to more abundance of calorie intake, now they are used to low calorie intake, where the body is adjusted to absorbing everything and saving everything or storing everything. Now, when the nutrients are abundant, still they tend to absorb everything and save everything, store everything, which leads to obesity and all the other related metabolic complications. Same goes with smoking and epigenetics. Now, we know that smoking is harmful, is hazardous. However, part of this harm is done through epigenetics. Now, what is the problem? The problem is just not harm to you alone, that these changes may be handed down along the line in the future generations as well. So, as in the case of maternal malnutrition, fetal exposure to smoker may produce short-term metabolic changes that compromise the long-term survival. Then there's another interesting theory, what we call environmentally induced epigenetic drift, where a study has demonstrated that monozygotic twins experience an epigenetic drift as they grow older, and depending on the environment that they get exposed to and their habits. And this, talking about the environment, we know that pollution, air pollution and other pollution causes serious cardiovascular risks. However, this risk may be through epigenetic alteration. I'm sure some of you have wondered that why you keep on gaining weight while your skinny friend, who eats way less than you do, doesn't gain weight. Now we live in an era of a obesity epidemic, and this obesogenic environment that we live in obviously plays a part. However, there is a substantial variation in how people respond to the same environment. When we talk about obesity, there have been studies have identified many DNA methylation or DNA epigenetic changes in the DNA in obese personnel. But there's a small problem. What we have identified is there's an epigenetic change in the genome, but we don't know which came first, whether it's kind of a which came first, the chicken or the egg came first scenario, whether the epigenetic change came first or the obesity caused the epigenetic change. And talking about cancer, now when we talk about cancer, we talk about genetic alteration, but cancer is much a disease of dysregulated epigenetic alterations as much as it's a genetic dysregulated genes. And it has been found that DNA methylation remains one of the major hallmark changes in most cancer types. So when summarizing, let me ask these questions again. Are epigenetics important to us as clinicians? I certainly believe so, because it is involved, it seems to be involved in many non-communicable diseases that we fail to manage or fail to diagnose. Can we alter our own epigenetics? Or does the environment we live in, our habits, alter our epigenetics? I certainly believe so. And can we alter our epigenetics through medication? That's definitely something to look forward for. But as we discussed, epigenetics was all about certain chemical changes to our genome. We do chemical changes to our body all the time. Maybe this is something we definitely have to look forward for. Are epigenetics inherited? The questions, these condition answers to these questions, definitely yes. So, little peek into the future. Epigenetic treatment for non-communicable diseases. We saw that there can be many epigenetic changes when you talk about non-communicable diseases. What if we can change it? We have a very long way to go. However, it's something I would definitely look forward for. What about predicting non-communicable diseases? What if we can identify through family history, if we can predict by looking at the epigenetics of a person that this person will get or might get a certain condition? Definitely it's a possibility. What about treating congenital diseases? By switching on or off a particular gene artificially, we may able to control congenital diseases. So the opportunities scope is limitless. I'd like to thank the College of Physicians and my past and future past and present trainers and my mentors. And thank you all for listening. Open for any questions.
Thank you, Dulanga, for that interesting lecture. And it's open for questions. First, from the physical audience, are there any questions? Can we have the mic?
Thank you, Dulanga. It's very interesting and excellent lecture. What about the advantages that we have in changing the epigenetics? Are there any fears in future? Is there any way that the future generations can alter the genetics and genetic genotype, changing the environment and change the landscapes, anything like that?
Yes, sir. There have been theories where say that the one problem with changing epigenetics is that it get inherited. If we change epigenetics depending on the current environment we live in future generations, it might be a completely different environment that they live in. So we can't predict how these epigenetic changes will act in the future. It's definitely a question that has been made.
Any other questions from the virtual physical audience? Yeah, anybody from virtual audience wants to have questions? In absence of any other questions, Dulanga, it's my pleasure to present you with the certificate of appreciation. And we are very thankful for your presentation.
Thank you very much.